Hi everyone, I'm Kate from the University of Queensland, and today I'll be sharing with you some really fascinating research about the gestational physiology of the short-beaked echidna. The short-beaked echidna is a fascinating species. While classed as mammals, they possess this weird yet wonderful mix of both mammalian and reptilian features. So while they lay eggs, just like reptiles do, the eggs then hatch to reveal altricial young that then lap up milk from teatless mammary glands, just like marsupial mammals. Despite this fascinating biology, we know very little about the reproductive physiology of the short-beaked echidna. So little, in fact, that if you open a textbook on reproduction in mammals and open to the monitoring section, you'll find one or two sentences stating very little is understood about reproduction in the short-beaked echidna. This is largely due to the difficulties in studying these animals in the wild and the fact that they have been very difficult to breed in captivity. However, with recent success in breeding echidnas in captivity at Currumbin Wildlife Sanctuary, where echidnas can now be bred routinely, we have been able to collect serial plasma samples from echidnas from mating through to egg lay, so that we can now potentially start filling in some of these gaps in those textbooks. The primary aims of this study were to characterise the gestational physiology of the short-beaked echidna, and in addition, to map changes in pouch morphology alongside changes in progesterone concentrations. For this research, we sampled 6 female short-beaked echidnas, 3 during the 2018 breeding season and another 3 during the 2019 breeding season. At the beginning of each breeding season, the females were paired with a male, and these pairs were monitored by video surveillance monitoring system to observe mating, egg lay, and incubation behaviours. Plasma samples were collected 3 times a week, and because the pouch is temporary, we monitored changes in development during these times of plasma collection, and ultrasonography was performed regularly to confirm gestation. Finally, once all the plasma samples were collected, these were then analysed for progesterone using enzyme immunoassay. Overall, 5 of the 6 female echidnas studied demonstrated periods of elevated progesterone concentrations, and of these 5 females, 7 gestational periods were observed, with 2 females having a second gestation period. However, in addition, we also observed 2 females to have a second increase in progesterone concentration that was not associated with an egg. As demonstrated on the composite graph, which includes all progesterone profiles regardless of egg, each progesterone profile was remarkably similar. Progesterone concentrations increased within 5 to 6 days of mating and reached peak levels within 6 to 15 days. From peak levels, progesterone declined over 4 to 11 days before egg lay, which consistently occurred 1 to 2 days before baseline levels resumed. From confirmed mating, we observed gestation to be consistently 17 days, and the incubation period was 10 to 11 days. In addition, we've been able to define the luteal phase of the estrous cycle. So briefly, the estrous cycle is a cyclical pattern of hormonal activity that allows females to have a period of sexual receptivity. It consists of 2 distinct phases, the first being the follicular phase, where eggs are recruited and estrogen is the dominant hormone, and the second phase being the luteal phase, which occurs following ovulation or the release of an egg, which is ready for fertilization, and this phase is dominated by the hormone progesterone. Of the 9 cycles observed, we found the luteal phase to be 14 to 16 days, regardless of whether an egg was laid or not. In terms of pouch development, we only observed development to occur during periods of gestation. In pregnant females, pouch lateral margins began to swell as progesterone concentrations increased. However, maximum pouch development was observed as progesterone returned to baseline concentrations. By the time of egg lay, the pouch was completely closed, as if drawn together by a drawstring, as shown in image J with the red outline. However, it should be noted that some regression is observed while the echidnas are under anaesthesia, so the tightness of the drawstring is not 100% clear in this picture. Our results have significantly enhanced our understanding of short-beaked echidna reproductive biology. We have confirmed that progesterone functions in the short-beaked echidna to maintain gestation, and that gestation does not extend beyond the luteal phase, which, if you remember, is the progesterone dominant phase of the estrous cycle. This feature is quite common in marsupial mammals, And similarly, the neonates of both echidnas, post-egg hatch, and marsupials are at a remarkably similar stage of development, both being attritial and requiring an extended period of lactation for further development. By confirming mating and egg lay, we have been able to confirm that gestation in the short-beaked echidna is 17 days long. This is shorter than the currently reported range of 20 to 24 days, which has been based off estimations and observations of wild populations. It is yet to be confirmed whether ovulation in the short-beaked echidna is spontaneous or induced. So while spontaneous ovulators will ovulate regardless of whether there's a male present or if they've been mated, induced ovulators do require to be mated or the presence of a male. However, we did observe 2 non-pregnant luteal phases when no matings were observed, there was no pouch development, ultrasonography failed to detect an egg in utero, and there was no incubation behaviour observed for these females. It's possible that these females had an anovulatory cycle, meaning that no egg was released at the time of ovulation, or maybe the egg was lost in the early stages of development in utero. Another possibility is that they had a spontaneous ovulation and just weren't mated at the right time. Given we have some evidence for induced ovulation and these non-egg related luteal phases, it's quite possible that the short-beaked echidna is both a spontaneous and an induced ovulator, which is quite common in many feline species and rabbits. In terms of pouch development, the initial swelling of the pouch lateral margins coincided with the increasing progesterone concentrations, but the maximum pouch development and maximum swelling of the lateral margins was associated with the decline in progesterone concentrations. So it's not quite clear if progesterone does play a role in the pouch development at this stage. It is thought that hypertrophy of the underlying mammary glands may contribute to the development of the pouch, which is presumably controlled by prolactin, as it is in other mammals. However, the ability of the pouch to close in a drawstring manner may not be under hormonal control, but perhaps mechanical control, as we observe the pouch to regress quite rapidly following egg removal at day 2 or 3 of incubation. I'd like to thank the many people involved in this research, starting with Currumbin Wildlife Sanctuary and all the zookeepers and veterinarians and vet nurses that help out with this research. Of course, my PhD supervisors, Stephen Johnson, Marilyn Renfrey and Tamara Keeley, and wouldn't be possible without funding from the Australian Research Council. Thanks for listening and happy to take any questions.